Hey, so welcome to lecture 30. And in the last class, we looked at cell-cell and cell extracellular matrix interactions. And so for cell-cell, we had a couple of important ones that were mediated by what are called coherins. And depending on what type of coherin it is, it will associate indirectly, so via these adapter proteins, to either the microfilaments, which are made of actin, and so these are called adherens junctions, or by a different set of adapters to intermediate filaments, and these are called desmosomes. Tight junctions form a ring all the way around the cell, and these can prevent the flow of materials between the cells, or at least limit their flow, and also, for example, limit the movement of proteins to within sort of one region of the membrane. Adherens junctions can contribute to that as well, so we can set up polarity in the membrane. And it's not shown on this diagram, but these also associate indirectly with cytoskeletal elements. Gap junctions, we form a direct link between the cytosol of two cells. It's a very narrow channel that forms, and so only small components like ions, for example, single amino acids can move through, but like proteins, nucleotides, these can't move through. So it's not for resource sharing, but rather it's for sharing information. And then we talked about interaction with the extracellular matrix. So the extracellular matrix is produced by the cells, and it has protein components, what we call proteoglycans, which are a little bit of protein and a lot of carbohydrate. And then there are adhesive molecules, which are glycoproteins, meaning that they're protein with a little bit of sugar component. And what we're seeing here is the interaction between the adhesive protein, which in this example is called laminin, and it interacts with the collagen and also with an integrin receptor. And so the integrin receptors, like we saw with coherin mediated, they can associate either indirectly with the microfilaments, in which case they're called focal adhesions, or indirectly with the intermediate filaments, in which case they're called hemidesmosomes. And we talked about how integrins are important signaling molecules that are capable of receiving signals from outside the cell, but also receiving signals from inside the cell as well. So they have this inside out and outside in signaling capability. We then were looking at the epidemiology of cancer, and so if we look at, say, Canadian statistics, and we look at how people die, most people are going to die of circulatory diseases and cancers. Okay? And so this is typical of a population that lives a long time. Younger populations or places where there's a lower life expectancy, generally what we see is that more people are dying of infectious diseases. And so this is one of the reasons we're interested in studying cancers and circulatory diseases, because they do affect many people, older people in the population. All right, so where we had left it off was we were talking a little bit about how geography affects cancers. And so this could mean a few different things. It could be sort of cultural differences. So, for example, maybe how acceptable are things like smoking or drinking or things like that. What about diet? These can all affect cancers. It could be something physical with the geography. So there might be some toxin, and maybe there's heavy air pollution. And so, of course, that can affect cancer rates. But geographically, there could also be a genetic component. So there may be an accumulation of certain mutations within a population or a community geographically. This first slide here is showing us that it's not solely genetic, because if we take someone from Japan and they move to California, their rates of cancers alter. So the occurrence of them getting stomach cancer drops, but for colon cancer increases. And so, of course, there's been no genetic change by moving to a new country. So this is indicating that there's either some cultural difference or actual physical difference in the geography itself or the place itself. And that was where we got to in the last class. Any questions from that? No? Okay. So here we are looking at a family tree. In this family tree, females are represented with the circle, males with square, and if someone has a slash through them, that means that person is deceased. It doesn't mean the person died of cancer, okay, it just means the person died. If the shape is blacked in, it means that that person at some point developed cancer. Again, it doesn't mean they died from the cancer. They may have fully recovered, but it means they developed cancer. And if we follow this through, it really, we can see that it looks like there is a genetic component here. Because, we have a lot of people in this family tree developing cancers. And so people can inherit a predisposition towards cancers. But I want to make this clear, you don't inherit cancer itself, for reasons that we'll look into later. These might be things like, for example, BRCA1 or BRCA2, you've maybe heard of. These are DNA repair genes. And so if you have a mutation to a DNA repair gene, we can imagine that might make you more likely to develop cancers. So BRCA1, this is the mutation that Angelina Jolie carries, for example. And so this was quite famous a few years ago. Her mother died of cancer many years ago. She found out she was carrying this mutation, and as sort of a preemptive approach, she had her breasts and ovaries removed. So because having a BRCA1 mutation, you have higher rates of cancers there. So some people may have heard of that. And there are many other genes that can be mutated that can increase the likelihood of cancers. But again, you're not inheriting the cancer itself. Oh yeah, so what's the weird thing about this particular family tree? Someone that's into genealogy. There's something that you shouldn't see in a family tree here. Yes? Yeah, so we got a couple of instances here of anyway. There we go. So, moving right along. So our cells are damaged all the time. And we pick up, it's estimated, something like billions of mutations every day. We, however, have very robust repair mechanisms. And so almost all mutations are fixed. Okay? If we're going to develop cancers, there's sort of two defining properties. The cells reproduce without the normal restraints or regulation, so they're not listening to those signals we've been talking about. And then they can invade or colonize other tissues. There's two types. We can have benign growths that are non-invasive, and malignant, which are invasive. And cancers generally contain or retain some of the hallmarks or the features of their parent tissue. And the example we looked at in the last couple classes was intermediate filaments are often retained. This, again, can be beneficial for helping us to treat a cancer if we know what the parent tissue is. So, amazingly, a cancer likely arises from a single aberrant cell. So one cell picks up, accumulates enough mutations that it now switches to become a cancerous cell. One of the issues is that the cancers aren't detectable until they get to a certain size. And the estimate that's here is about 1 billion cells. This is a problem because the earlier we detect a cancer, the better the prognosis, the better the chances of treating it are. Okay? And so a lot of research, cancer research, actually goes into detection. And some of them look at what are called biomarkers. And so a biomarker is some molecule that's produced that indicates or it's specific to a cancer. And it's something that we can detect easily. So this might be, the famous ones are the drop of blood ones, where you get a blood test, they take blood, and by analyzing the blood, we can detect cancers. And there's a lab at UVic that's exploring this. Actually, there's a couple exploring this. Elizabeth, I can't remember her last name now. Warren, the Theranos person, is on trial right now. They set up this huge company, took a whole bunch of money from this, and had no idea how to do biomarkers. They weren't doing it at all. So this is a big case going on in the States right now. And unfortunately, it's really stained the biomarker research industry because now investors clump it all together as kind of garbage science. So it was quite a terrible blow to the biomarker community because these are valuable tests. If we think of cancer tests, many of them are very unpleasant, okay? And they're invasive. You often need to go to a specialist to get these done. They can be expensive. And so these are all barriers to early detection. Also, the tumor has to be a certain size usually to be detected. In contrast, if we could find these from, say, a blood test or a urine test or a stool sample, people are more likely to get tested, and the sensitivity may, in fact, be higher for these. Okay? And so this is why we hear a lot about biomarkers. And the bottom one is just that there's many different types of cancers, but we'll explore that as we move on. So because it's due to an accumulation of mutations, this is why they don't tend to develop until later in life. We also see an effect in that once a cell starts to accumulate mutations, it becomes more likely to accumulate more mutations because, for example, many of the DNA repair pathways become damaged. And so the cells are not as capable of fixing their DNA, and they're more likely to accumulate. So this could explain why we see this exponential increase in the number of cancers with age. Okay, so we have age on the x-axis, incidence rates per 100,000 on the y. So the number I had was that every single gene we have will be mutated about 1 million times. I think the textbook lists 10 to the 8th. Each gene in our body will be mutated that number of times. This is, of course, almost always fixed. Okay? And I think when they're looking at those numbers, they mean across all cells. So gene A won't necessarily be mutated 10 to the 8th times in each and every cell, but if we were to look at all of our, 33 trillion cells, it would be mutated that number of times over our lifetime. And those numbers aren't really important, except that they're saying, yes, we incur a lot of DNA damage, but it's almost all repaired. And so the tumors progress, and so they start out with sort of mild changes, and then these become more and more, I'm going to call them catastrophic, until they become cancers. So we have two types of cells, somatic and germline cells. The germline cells are the egg and sperm. These are haploid, have a single copy of the DNA. The somatic cells are the cells that are cancerous. And so these are all of the other cells that we have. If someone has a germline mutation, okay, so in this example, the sperm is shown as pink, and so this is carrying a germline mutation. This means every single cell in the body will carry that mutation because we have a paternal and a maternal copy of all the genes. When this person forms gametes, remember gametes are haploid, so it means half of their gametes will carry the mutation, the two pink ones here. The blue ones are non-mutated, okay, because that's the copy that is, in this case, the maternal copy. In contrast, if we look at a somatic mutation, both the egg and the sperm are fine, and so the gametes formed here, none of them carry the mutation. But what happens is one or a little cluster of cells pick up a mutation. And remember, as we grow, our cells are always dividing, making daughter cells, and those daughter cells will now each pick up the mutation. And so you end up with a patch or a cluster, a small area where you have these somatic mutations. And so for cancer, we get a large accumulation of mutations in the same somatic tissues. And incredibly, it's probably on the order of 50 to 75 mutations are required for cancer to occur, depending on what the mutations are. And this is why I was saying in the last class that every cancer is unique, because they won't accumulate the same mutations. There's certain genes that are more likely to be mutated, and we'll talk about the prevalence of some of those. At the top here, we have a progression, just an example of a progression. And so we start with something called APC. This stands for adenomatous polyposis carcinoma. Don't worry about it. The only reason I'm pointing this name out is when we talk about the cell cycle next week, we're going to talk about something called the anaphase promoting complex. And this is not the anaphase promoting complex here. Here's a RAS mutation. Remember RAS, our old friend RAS, may be involved in up to 50% of cancers. Here's a SMAD4. Look at that, a TGF-beta mutation. P53, okay, this is a tumor suppressor we'll look at next week. And then other mutations. And what we can see is that these progress from a benign tumor to, at the end here, a metastatic tumor, one that picks up and can move to new locations and is invasive to the tissues. So it's kind of like evolution. The cells pick up mutations, either genetic or epigenetic, and then there's a selective process. And the cells that can grow and replicate in that environment tend to predominate in that environment. As a tumor grows, and I guess something we need to think about is it's a harsh environment. So within the tumor itself, once the tumor starts to form, we may have lack of circulation, so we may be lacking things like oxygen or nutrients. There's the immune system to deal with, and so there are all of these selective pressures on the cancer. And what we see then, and the parameters that affect it, are mutation rate, number of reproducing individuals, rate of reproduction, and selective advantage. And these are the same things we see in a population driving evolutionary changes. So tumor is any abnormal growth or mass of tissue which exceeds and is uncoordinated with that of the normal tissue and persists in the same excessive manner after cessation of the stimuli which evoke the change. There we go. What does that actually mean? It means these things grow without the usual signals for growth and replication. So they don't follow the cell cycle. Again, we can have benign or malignant. So benign are confined to a specific area, generally not considered dangerous. A good example of this that most people are familiar with is when you have moles on your skin, you know, the ones that are nice and round, and they say, don't worry, that's benign. You see those charts of what's dangerous and what isn't. Whereas malignant, these become capable of invading surrounding tissues, and they may even be metastatic, in which case they can enter the bloodstream and move to distant parts of the body. So we can use a few terms. So carcinomas are epithelial in origin. Okay, so skin, brain, breast, cancers. Sarcomas are for connective tissue, so cartilage, bone, muscle. Lymphomas are lymphoid tissue, okay, so thymus, bone marrow. Melanoma, we can utilize this term for pigmented skin. Okay, so again, kind of carcinoma. Hepatoma for liver carcinomas. Again, the liver accumulates or processes toxins. The blood filters through the liver, so these are often places where we get cancers. Leukemias are the white blood cells, cancers of the white blood cells. So you get a massive proliferation of white blood cells. Excuse me. And blastomas are a class of childhood tumors. So there's different types of cell growth, and the top two are in response to a stimuli. So this, oh, question.
Sorry, I don't know if it's a dumb question, but could you give an explanation the difference between the carcinoma and the melanoma?
So the melanoma is just a specific type of carcinoma. Okay. Yeah, just like the hepatoma is just a type of liver, a type of carcinoma in the liver. I think it's to do with the prevalence of occurrence. So melanomas are fairly common, and so they have a specific name. I would imagine all the different epithelial carcinomas do have different specific names, depending on the tissue. Yeah?
Is blastoma only for children?
That I'm not sure about. I'm not sure about, sorry. So in response to stimuli, cells can do a couple of things that cause them to increase in size or mass. If they increase in size, we call this hypertrophy. But they maintain an organization, hypertrophic cells. Hyperplasic, we get an increase in the number of cells. So we're still increasing the size of whatever this tissue is, but they maintain their organization. And if a signal is removed at this point, the tissues will return to the normal, what they normally look like. So the left-hand side is the normal side, good side, bad side. Dysplasia, what happens is we start to see some level of disorganization. It looks like there are some hypertrophic cells in here, there's some hyperplasia, but we're getting disorganization. And at this point, removal of the stimuli may or may not lead to a reversal. And so this is sort of an in-betweener. In contrast, the neoplasia, these are no longer dependent on the stimuli. If you remove the stimuli, it won't make any difference. They can't revert. So this is what we call a tumor, a neoplasia. Again, what we're looking at here is comparing benign and malignant. And again, for me, I always think of these in terms of skin cancers because they're visible, and we kind of had this drummed into our heads about checking your moles and the features of your moles and whether or not that meant it was an okay mole or a problematic one. And we can see the reasons here. So some of them, of course, you can't just see. So things like the nuclear size. Malignant cells have larger nuclei and a higher ratio of nuclei to cytoplasm. So the nucleus takes up more of the cell. The shape in benign is regular, but in malignant, the shape is pleomorphic or irregular. So again, think of a mold. They always say if it's got nice, it's rounded and it has nice borders or margins, it's okay, but when they start to spread or take on a weird shape, that becomes problematic. The mitotic index is a relative number of dividing cells. So the actual process of replication takes the same amount of time in a cancer cell and a normal cell. So they can't replicate the DNA faster or anything like that. But what happens is they replicate more often. So these gap periods we're going to see later are shorter. And they have a higher mitotic index, meaning instead of like 10% of the cells replicating at any given time, maybe 25 or 20% of them are replicating at any given time. And so that's what the mitotic index is. The tissues are disorganized in a malignant tumor and poorly differentiated. So they, remember, differentiation is when a cell becomes specialized. So we talked about epithelial cells, and then we talked about a tumor. They go through the EMT, so they go back to a mesenchymal form or a more primitive form. And the boundary, again, poorly defined in a malignant tumor. So in a normal sort of growing cell, what happens is we get division of a cell into two cells, and only one of those cells retains the ability to replicate. And so we just end up with replacement along the way. So one cell dies, a new one comes in. One cell dies, a new one comes in. That's lost in a tumor. Okay? And so we get this increased mass in the number of cells, and therefore the size of the particular tumor. Okay? And so this can be due to a failure of differentiation, meaning the cells maintain the ability to replicate, or a loss of cell death. So cells undergo programmed cell death to maintain the cell numbers in tissues, and that can be that process can be lost. This is odd and maybe a bit counterintuitive, but that there's not a correlation between the cell division rates within a tissue, the normal cell division rates, and cancer. So for example, in our small intestine, we have very rapid rates of cell division. And so, as we see here, something like 40 kilograms per year of gastrointestinal cells are produced. Brain cells almost never divide, almost never replicate, and yet brain tumors are reasonably frequent. Okay? And so, oddly enough, there's no correlation there between the rates of replication and frequency of cancer. There's a couple of other sort of things here that people can die from a tumor weighing less than 5 kilograms, actually significantly less than 5 kilograms. And again, the earlier we detect these, the higher the likelihood is of a successful intervention. So the sort of 4 things that are going to cause cancer are viruses, infectious agents, breakdown in immunity, and genetic mutations. So you don't have a strong background at this point in genetics, but we can keep it reasonably simple, just dealing with the concepts that we've talked about already in the class. And so one of the things that some viruses are capable of doing is integrating into the genome. And that means the viral genome actually becomes part of your genome. It actually inserts into it. And this can disrupt the regulation of genes, or it can disrupt the genes themselves. So if it inserts into that region we talked about, that coding region, that can prevent that protein product from being made. Or if it inserts into the promoter region, the one that regulates it, that can affect the regulation. Viruses can actually carry genes that contribute to cancers as well. So they don't actually even, so it can be that the viral genome itself contains genes that promote cancer. So it's not necessarily just disrupting, in the case of humans, disrupting a human gene or a human promoter. The virus itself can carry genes that can promote cancers. And these genes that promote growth are called oncogenes. So oncogenes promote growth. So we can activate or introduce oncogenes, or, so these are growth, or we can inactivate important genes. These might be things like DNA repair genes, for example. So here's an example. This viral DNA is carrying an oncogene with it. And this is a gene that is tightly regulated, normally in humans, and will only be activated by the MAP kinase pathway. This is a protein called MYC. So this is one of those mitogen-activated proteins that are transcription factors activated by mitogens. This virus just happens to be carrying a copy of it around, and it's an unregulated copy. So this inserts into the host genome, and now there's a whole bunch of MYC being produced when there shouldn't be. It's being produced even in the absence of other cellular signals. Okay? This doesn't necessarily cause cancer because we have a lot of redundancy built into our signaling pathways, right? And a lot of feedback. Remember we talked about how our signaling pathways work hard for feedback and resetting the baseline. And so a single mutation like this can be overcome, but it can contribute to those one of the 50 or 70 or so mutations that are required for a cancer to form. In this example, the viral DNA isn't anything special except that it can integrate into the host. And here it's disrupting whatever the regulatory region. So this would be the regulatory region. The reg. Reggy. Okay, so the regulatory region, we're disrupting it here with the viral DNA. So now the MYC it doesn't behave like it should. And in this example, we're seeing we get excess production. So that's one way that viruses can promote cancers. Certain viruses, such as Epstein-Barr virus, EBV, and HIV, human immunodeficiency virus, these suppress our immune system. And in fact, our immune system is capable of recognizing cancers. And it can destroy them before they can destroy aberrant cells before they become cancerous, and they can work to battle against tumors. But if their function is suppressed, we see an increase in the numbers of certain types of cancers. And we'll come back to that in a minute. Infection can lead to inflammation. Chronic inflammation, so temporary inflammation is good. That's your immune response responding to, say, a bacterial infection. And it's part of the process. And it's beneficial in the short term. Long-term inflammation or chronic inflammation is not good. And this has a lot of negative outcomes for people. And chronic inflammation seems to contribute or increase the probability of certain cancers if tissues are inflamed all the time. Okay? And so this would be like the hepatitis B and hep C are kind of well known for this. So if you have hep C or B infection, there's a higher likelihood of certain cancers. H. pylori, we have this, I think it's the next slide, is a bacterial infection that causes gastric ulcers. Okay? And there's a correlation, I'll go to that slide here, there's a correlation, a slight correlation with increased risk of cancer with people that have stomach ulcers. Again, this is a correlation, not necessarily a causation. And other nasty cancer, or nasty viruses actually directly promote tumor formation. So things like HTLV, which is human T lymphoma virus, I think, and HPV, which is the human papillomavirus, these can promote cancers. We'll come back to HPV, human papillomavirus, either on Tuesday or Wednesday's lecture, and we'll look at what it does, but it actually directly promotes cancer. It shuts off tumor suppressor genes. So immunity plays a role, all right? And the immune system can recognize cancerous cells. A cancer that progresses often has developed in such a way that it hides from the immune system, or the immune system can't effectively access it. Okay? And so a breakdown of the ability of the immune system to recognize the cancer can contribute to malignancy. Just a bit of evidence for this. Many cancers infiltrate the lymph nodes. Okay. The lymph nodes are basically where the immune system hangs out, I guess. So we have a number of these around the body, and lymph is pumped through, and this is where we deal with things like infection, and this is, we get accumulations of cancers in them often. If we look at tumor-associated proteins, antibodies can be raised against these. And then the last one is people that have compromised immune systems have a much higher likelihood of certain cancers. The statistics that I have are that, for example, for HIV, one of the things that was noted early, sort of in the 80s, was that there was a very rare type of cancer called a Kaposi sarcoma. Oops. Kaposi sarcoma. It causes facial lesions, and it was found that these were developing at a much higher rate than people that had HIV infections. And it was 500 times, people were 500 times more likely to get Kaposi sarcoma, for example. And this was cancer that was associated with AIDS at the time. In addition, as we said, other types of viral infections can lead to higher rates of certain cancers. And these are viruses that compromise the immune system. On the plus side, many of you have maybe heard of CAR T cell therapy. So this stands for chimeric antigen receptor. So what we do is we take the T cells from the patient, and some of you may have been to Yvonne Stevens' talk. She talked about this. Her company Lyle is working with this in Seattle. She gave our first talk this year. And what we do is we engineer them so that they have a receptor on the surface, this chimeric antigen receptor, that recognizes something on the surface of a tumor. So we engineer this onto this T cell. We then grow up the T cells. Keep in mind, these T cells have come from the patient. So you don't have to worry about matching the HLA, which is what you normally need to do for organ transplant or blood transplant. You have to make sure you match the surface antigens so your body doesn't recognize it and attack it. So these start from the patient themselves. We engineer them to recognize the tumor, grow them up, and then infuse them back into the patient, and they attack the tumors. And these, there's been a lot of success with this. And so these treatments look as if they're very promising. They're still being developed, but they are set up now at BC Cancer. and at the Dealey Center. So they are set up to actually do this now down there. I've been through the facility. It's pretty amazing, and some of you might take courses with Dr. Brad Nelson or Dr. Julian Lum later in your career, and they'll talk more about that because they're the experts at the CAR T. So again, this wouldn't work if our immune system doesn't play some role in cancer development. So it is also a genetic disease, however. And so the genetics and epigenetics of this can be affected by a whole host of environmental factors. All right, so environment, behavior, diet, all of these things can result in mutations, and those mutations may contribute to development of a cancer. Again, every time there's a mutation, that mutation is either lost or retained, and this works much in the same way, as we said, that we see in the evolution of different populations. So there's some selective pressures in there. All right, so the three types of genes are oncogenes, tumor suppressors, and DNA repair. These are the main places where we see mutations. We can think of them, the oncogenes or the go genes, all right, like the accelerator in a car. Tumor suppressors are the stop, like the brakes. And then the DNA repair, of course, these are the genes that can correct genetic mutations, fix them. So we'll look at oncogenes. Oncogenes can trigger, definitely they contribute to the development of cancer. The first one discovered was what's called the Rous sarcoma virus. So at the top here, we can see this is a murine leukemia virus. And viruses often have these what are called the GAG, POL, and envelope genes. These are what we call structural. These are for replicating the virus, and these are for the envelope around it. And the first oncogene that was discovered was a virus that caused cancer in chickens. And when the viral DNA was analyzed, it was recognized that they had an additional gene on them. And this is one we looked at earlier when we were talking about domains called SARC. This is a tyrosine kinase. It's not a receptor tyrosine kinase. It's cytosolic, but it's a signaling protein. And what happens is that it promotes growth. And so the chickens that get infected with this is what we saw at the start of our viral section. They get this oncogene, which is unregulated and is produced all the time. The oncogenes play a role in the cell cycle. We'll spend time on the cell cycle in our, I think it's the last lecture we'll be doing. And so don't worry about this now, we'll come back to it. So there's a little bit of terminology, and sometimes the cellular version of an oncogene is sometimes called a proto-oncogene. And so, of course, we have lots of oncogenes because our cells replicate all the time. So we have to have oncogenes that will contribute to that or promote that. And so sometimes the normal regulated cellular versions are called proto-oncogenes. When they become mutated so that they gain activity, and so that now these oncogenes, either through a promoter, well, we'll see how they become promoted to oncogenes in the next step. Then they would change the name to oncogene. I think this terminology is used less commonly now. And I've talked to a few people about this, and they all seem to be in agreement that generally now, all the versions they call oncogenes now, rather than separating into proto-oncogene, which is normal, and the mutated form, which is an oncogene. So that gets a little bit confusing when we're looking at things. And so I'll try to describe when I'm looking at these exactly whether it's in the normal form or not. So here's a few ways we can convert from a proto-oncogene to an oncogene. So these are all genetic things that happen. So the proto-oncogene, remember here, this is normal regulation. So we might end up having a hyperactive protein, so one that is regulated normally. We make the right amount of it, but it's overactive. We may duplicate the gene so that we're getting multiple copies. We may get disruption of the regulatory regions, okay, either through natural events or within the cell events or through viral insertion. And then we can get other forms of the hyperactive protein. And this promotes then the gene from a proto-oncogene to an oncogene. And one of the best studies of these is what's known as the Philadelphia chromosome. And so the Philadelphia chromosome is that on chromosome 9 we have what's called the C-ABL gene, and this is the oncogene. And it's tightly regulated. There is a reasonably common translocation. Translocation is when one part DNA is moved from one location to another one. And in this case, it's moved from chromosome 22. And it's something called the BCR gene. And the BCR gene is on all the time. And the BCR translocates and makes what's called a fusion gene, so that we have the BCR regulatory domain. And we make a protein that's BCR, and then the reading frame actually includes ABL. So it makes one protein that's both BCR and ABL, but a single polypeptide. But the ABL still acts as an oncogene. And since BCR is produced all the time, this BCR-ABL fusion gene is now always promoting growth. And this is found in 95% of chronic myelogenous leukemia cases. And when this was recognized, this was the first time that a drug was designed specifically for a target. So this was the first example where instead of just looking for effects and sort of this scattershot approach to finding drugs, they had this target and they looked for that target specifically. And we're going to stop there, and we'll see everybody next week.